DNA damage and repair. So we have seen the process responsible for damage of DNA. Now how this damaged DNA is repaired in nature inside the cell, that we will see here. We have seen DNA damage can occur due to exposure to the UV radiation, X-rays, or exposure to certain chemical substances. Now how these damaged DNA is repaired. So repairing mechanism, we can call it as a repair mechanism in general. So how it happens in eukaryotic cells as well as prokaryotic cells, that we will see with the help of some of the theories. So first theory is SOS repair, or also called as a recombinational repair. And another name for this theory is error prone repair. So in this diagrammatic representation, SOS repair, how it occur, that is shown. So SOS repair is a mechanism of DNA repair which is induced in Escherichia coli by ultraviolet radiations or by many other agents which either damage DNA or inhibit wrong way replication. The SOS DNA repair mechanism ensures survival of the organism as it directs wrong way DNA synthesis to right way. This type of DNA repair responds to extreme stress, and therefore it is called as a SOS repair. Meaning of SOS repair: SOS is the urgent call for help to repair the damaged DNA. So that is the meaning, literally meaning of the SOS, urgent call for help to repair the damaged DNA. In other words, we can try to understand this SOS mechanism of repair, the mechanism by which Escherichia coli cell repair its large amount of DNA damage, and that is called as a SOS repair, also called as a indivisible DNA repair. SOS repairing mechanism is also known as a recombinational repair of DNA. SOS actually consists of a group of at least 15 genes which are involved in repairing of damaged DNA. They are induced by protein called as REC protein, REC. REC A protein is responsible for this thing, and SOS repair is a very efficient mechanism to repair damaged DNA, but however it has a prone or tendency to insert mismatched bases while repairing the DNA, and thus SOS repair is error prone or tendency of doing errors, and it introduces additional mutations or additional DNA damage even. The another method of DNA repair is photoreactivation. We have seen related to the SOS in brief. Now photoreactivation, also called as a photo repair. So how it happens, that is shown over here in this diagrammatic figure. So photoreactivation is also a type of DNA repair mechanism. In this mechanism, ultraviolet induced pyrimidine dimers, maybe thymine or cytosine dimers, are reversed to pyrimidine monomers, that is T and T or C and C, on exposure to the visible light. So dimers produced due to ultraviolet rays is reversed, and thymine or dimer gets separated into monomer like this. Many kinds of bacterial cells and yeast cells possess this efficient mechanism of DNA repair, that is photoreactivation, for repairing the DNA damage caused due to the ultraviolet radiation. So how it happens in this mechanism, when cells are exposed to heavy dose of ultraviolet light, the DNA pyrimidine dimers are produced, and these pyrimidine dimers are like TT. So you can see here this TT dimer or cytosine dimer, CC dimer, where T is linked with adjacent T, forming the thymine dimer. And same is the case with C links with adjacent cytosine links with adjacent cytosine and forms a cytosine dimer. When a cell possessing such UV-induced pyrimidine dimer, then these cells, they are exposed immediately to visible light. So dimer is caused due to UV radiation, and then they are exposed to the visible light. In next process, what happens, or you can say blue light, out of visible blue light, the dimers are split into monomer pyrimidine bases, and thus the damaged DNA gets repaired to its normal state. This DNA repair mechanism depends upon exposing the cells to visible light following UV irradiation, and hence it is called as a photoreactivation. A special enzyme here is involved in this repair mechanism, and that is photoreactivating enzyme, and also called it is named as a proteolase. So this enzyme is activated by photons of visible light, and this activated proteolase breaks the bond between pyrimidine dimer. So like this, the activation of enzyme is due to the visible light, and thereby splitting of the dimer into the monomer and restoring the DNA or repairing the DNA to its normal state. Thus, photoreactivation is light dependent and enzyme mediated mechanism of light dependent and enzyme mediated mechanism of DNA repair. So this is the another method of repair of DNA. Next to that, we will go towards the third method of DNA repair, and that is excision repair. Excision repair. So this excision repair is diagrammatically represented over here. Before that, we will go towards the how it happens in case of photoreactivation. See here, it is a normal DNA. Now, due to exposure to the ultraviolet rays, the formation of thymine dimer over here. So in this region, thymine dimer is shown, and after that, when the DNA is exposed to normal or visible light, due to the blue light, the enzyme gets activated, and this enzyme is shown over here. So this activated enzyme, it is responsible for breaking down the bonds between the thymine and the formation of monomer and restoring the DNA or normal state of the DNA due to the removal of this dimer. So I think you might have understand how the mechanism of photoreactivation is. Now we will go towards the another mechanism of DNA repair, and that is excision repair. So here in this diagrammatic sketch, this excision repair mechanism is shown. So excision repair, it is also called as a dark repair or dark reactivation. Photoreactivation we have seen. Now here this process do not require light, and that's why called as a dark repair or dark reactivation, also called as a excision repair. Excision means to remove. So here part of the damaged DNA is removed, and that's why the process of repair is named as a excision repair. So see here how it happens. The method named as a excision repair or dark repair. So in this or by this type of DNA repair mechanism, DNA can be repaired. Some mutant strains of Escherichia coli are extremely sensitive to UV radiation, and UV induced dimers are corrected when these bacterial sensitive strains are stored in dark. See, some mutant strains of Escherichia coli bacteria, they are extremely sensitive to ultraviolet radiation, and when such sensitive strains of bacteria are kept in dark, DNA error gets cured or repaired. So form dimers, they are, or the dimer having part of the DNA is excised, and the DNA gets repaired. So such DNA error correcting mechanism, we can call it as a excision repair or dark repair. So how it takes place here, that we will see in brief. So here you can see in first figure you can see the presence of the dimer or formation of dimer due to extreme sensitivity to ultraviolet radiation in the DNA. Now when these sensitive cells are kept in dark, then what happens? The nick or you can say break on both the sides of the dimer having some other nucleotides. So nicks are made or breaks are made on both the sides, and this nick formation is due to the enzyme, and that enzyme is called as a exonuclease. Enzyme responsible for breaking the dimer on both the sides is called as a exonuclease. And due to formation of nicks on both the side of the damaged DNA, the here you can see this part is removed from the DNA, and later the gap produced is filled up by newly synthesizing DNA fragment, and the responsible enzyme over here is a DNA polymerase. So DNA polymerase is responsible for filling the gap created due to the excised thymine dimer here. And DNA ligase is the enzyme which joins the newly synthesized DNA fragment, and restoring of the DNA or repair of the DNA is here. So like this we can give the explanation for excision repair or dark repair or dark reactivation method of the DNA repair. So these are the methods to see the mechanism of DNA repair. So these methods can be asked in the examination. Out of two methods at least: excision repair or dark repair, then photo reactivation, and source repair. So these three methods we have studied in brief, and this completes the topic DNA damage and repair.